So what we did in this study is we took 11 patients with melanoma that were treated with anti-PD-1 alone or in combination with other immune checkpoint blockade. And we divide them first by responders and non-responders to therapy. And then what we did was isolating neoepitope-specific T-cells from samples from the blood and the TIL, so tumor-infiltrating lymphocytes, from these patients. And we were lucky enough to have samples from baseline and also different samples over the course of the treatment from all of these patients. So what we did was isolating the neoepitope-specific T-cells using a technology newly developed by a company that we were collaborating with. The company is called Pac Pharma. And using their technology, we were able to identify which are the antigens being targeted in these patients and which is the sequence of the TCRs targeting these antigens. With this, what we saw, the first thing that we saw is that in patients, both in patients that respond or do not respond to therapy, we are able to isolate neoepitope-specific T-cells that target mutations in the tumor. But what we see is that and then these responses are very immunodominant, which means that only a very small number of antigens are being targeted, both in patients with and without response to therapy. But the difference between these patients is that in the patients with response, these T-cell responses are polyclonal, meaning that there's different T-cells with different TCR sequences that target the same mutation, and also these different T-cells, these different T-cell response evolve over time with different T-cell clonotypes evolving and changing the frequency over time, but always consistently targeting the same mutations. While in patients that do not respond to therapy, we were able to isolate these T-cells, but they were not expanded over time, and we were not able to find them both in the periphery and in the tumor.